Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
What if there was a way to recover after a stroke that wasn't just all about, grinding through intense physical therapy?
Yeah,
Imagine like a pill that could help your brain kind of like relearn all those skills that were lost.
like a shortcut.
Yeah, exactly. Maybe not a shortcut, but an additional boost,
Right.
like a partner to the physical therapy. Well, this deep dive actually takes us right into that, looking at some really cool new research about this possibility, focusing on how do our brains actually relearn movement?
Yeah. How does that work?
Yeah, and there's this really surprisingly critical role of these specific brain cells. They're called Parvalbumin interneurons. So
Wow, that's a mouthful.
I know, right?
Parvalbumin interneurons.
Yeah, we'll get into more about that. But to bring you this, we've actually gone through a really detailed scientific paper and a super interesting health news article. So we've got all our bases covered today.
Yeah. So what we're going to try to do in this deep dive is really understand, the very specific neural pathways like the roadmap and then the molecular mechanisms that actually allow the brain to recover using rehabilitation after a stroke.
So like the brain's own recovery blueprint.
Exactly. Yeah.
That's a great way to put it.
And we're going to break down this recent study that aimed to kind of figure all this out with the ultimate hope that, this would lead to new medications that could actually help this whole recovery process.
Yeah, so the scientists behind this study, they had this like really specific question they were trying to answer. Could they actually find the exact brain circuits that are responsible for those improvements that we see with rehab after a stroke?
Pinpoint them.
Yeah, pinpoint them, exactly.
Yeah.
And even more interesting, could they then use that information to develop a drug, a medication that could basically mimic the benefits that we see from rehab?
Oh.
Yeah, and they were really focused on that time period right after a stroke, like those weeks and months after, because that's when the brain seems to be the most adaptable, and that's when rehab is usually the most effective.
Makes sense. So how do they study this?
Well, they did a really detailed study with mice. So what they did is they caused strokes in some of the mice, and then they put some of those mice through this like super intense repetitive training program for their front paws, which is fascinating.
Like tiny little mousy PT.
Exactly. It's basically like mouse physical therapy, and it was designed to mimic that same kind of rehab therapy that humans would go through after a stroke.
Oh, that's cool.
So they're really trying to replicate what happens in people, and then they used all these different techniques to actually watch what was going on in the brains of these mice.
Okay, so what do you mean like watch? Like how do you watch a brain working?
Yeah, so when we say watch, we're talking about using really advanced techniques. What they did is they had a few methods. First, they did behavioral tests where they looked at how well the mice could do things like reach out and grab an object, which is a pretty complex skill for a little mouse.
Yeah, especially after a stroke.
Exactly, and they called that the skilled reaching test.
Okay.
And they also looked at how steadily they could walk across this grid, which tells us about their balance and coordination.
Makes sense, yeah.
And then they use all these different brain imaging techniques, and these brain imaging techniques allow you to see which cells in the brain are active and how they're actually connected to each other.
Wow.
So they were really tracing these physical connections between the neurons, like those brain cells,
I see.
and they could even analyze which genes were being turned on or expressed inside these cells,
Oh, wow.
and how the connections between those cells, which are called synapses, how those were changing.
So like they could see the brain actually rewiring itself.
Exactly. They could see those physical changes happening. And then a crucial part of the study, they also tested how specific drugs impacted that whole recovery process in the mice. So that was like the final piece.
Okay, so the mice did that. They went through their little mouse rehab. Did it help? Did they get better?
Yeah, so the first thing is that the rehab program really did help those mice recover their motor skills.
Right.
So they got a lot of their ability back to reach and grab objects.
Okay.
And they could walk a lot better, too. So this mouse model is a really good way to study what happens in humans because it seems like the mice are recovering in a similar way to how people recover with rehab.
Makes sense. So they saw the rehab working. How did they figure out like which part of the brain was the most important for this recovery?
Yeah, that's the really cool part. So after some of the mice had actually gotten their motor skills back, the researchers induced a second stroke.
Oh no, poor mice.
I know, but this was a very, very precise, tiny stroke in a specific part of the brain called the premotor cortex.
Mhm.
And This premotor cortex is like the brain's rehearsal space for movements, like before you actually do them. So you're thinking about what you're going to do that's happening in the premotor
Oh,
cortex.
interesting.
Yeah, and they focused on a part of the premotor cortex called the rostral forelimb area.
Okay.
And what they found was that this second stroke in that very specific area completely erased the motor skills that the mice had just learned through all that rehab.
Oh, wow.
Yeah. So what that tells us is it's not just like general brain recovery, but this very specific area is super important for regaining movement through therapy.
Like the main control center for relearning movement.
Exactly. So then they looked more closely at that rostral forelimb area while the mice were doing rehab, and what they saw was really interesting.
What'd they see?
They saw that in the mice who were going through rehab, there was a big increase in the number of brain cells that were active in that area,
Oh, okay.
and also a big increase in the connections between those cells. So rehab was basically rewiring that part of the brain.
Like adding more connections, more roads between the different parts of the brain.
Yeah, more roads, and making those roads kind of like superhighways.
Cool.
Yeah, it's pretty amazing. And that brings us to those parvalbumin interneurons that we talked about at the beginning.
The tongue twister.
Yeah, exactly. So these parvalbumin interneurons, they saw that there was a big increase in the connections between those cells and the cells in the rostral forelimb area that were sending signals down to where the first stroke had happened.
Wow. Okay. So more connections between the rehab control center and the area affected by the stroke.
Exactly, yeah. And when we talk about connections, we're talking about those tiny little gaps between brain cells called synapses, where the cells talk to each other.
Right.
So more connections means stronger communication. And this was especially interesting because we already know that when the brain learns new things, including learning new motor skills, there are changes in these connections between different types of interneurons. So that was like one piece of the puzzle.
And these Parvalbumin interneurons specifically seem to be adding more of their connection points.
Yeah, and those connection points are called synapses. But the question is, are these parvalbumin interneurons actually causing the recovery? Are they actively involved, or are they just like watching it happen?
Right. So what do they find?
They found evidence that these cells are actually causing the recovery.
Really? Oh.
So what they did is they used this technique called chemogenetics. It's this way to inhibit or turn down the activity of specific types of brain cells, like a remote control for brain cells, basically. And in this case, they turned down the activity of those parvalbumin interneurons.
Clever.
I know, right? And guess what happened when they did that during the rehab? The mice didn't recover their motor skills after the stroke.
Wow, so they're definitely essential.
Yeah, so that tells us those cells aren't just bystanders, they're playing a critical role in this whole process.
So they're the key. What are they actually doing, though? Like, how are they making this recovery happen?
Good question. The study found that rehab actually increased the expression of certain genes and these things called plasticity markers in the parvalbumin interneurons and those stroke-projecting neurons in the rostral forelimb area. And these plasticity markers, they're like signs that the brain is actively rewiring itself and making new connections. So rehab was actually activating the specific circuit that involves the parvalbumin interneurons.
Okay, so rehab turns up the volume on these genes and markers, and that basically tells the brain to rewire itself.
Exactly, yeah. And they also found that the brain waves called gamma oscillations were involved.
Gamma oscillations.
Yeah, so the researchers found that in both the mice and when they looked at data from human stroke patients, that recovery was linked to more connections between neurons and these gamma oscillations. And parvalbumin interneurons are really important for regulating how neurons connect and also for generating these gamma waves.
What do gamma waves actually do, though?
They're kind of like the brain's rhythm for coordinating all that activity. And the parvalbumin cells seem to be like the conductors, keeping that rhythm steady so movement can be smooth. So you can think of it like this. The stroke messes up that rhythm, that synchronization, and then rehab helps to get it back on track, possibly because of the actions of these parvalbumin interneurons.
So it's all about getting the brain back in sync.
Yeah, that's a good way to put it. And they found that the stroke actually caused a decrease in these signals that the parvalbumin interneurons send, and rehab restored them. So that's even more evidence that these cells are really important for getting that balance back.
That's really amazing. So we know rehab is good. This rostral forelimb area is super important. And these parvalbumin interneurons are driving the recovery. So does this mean there might be new treatments in the future?
Well, that's where things get really interesting. They actually tested two drugs in this study. One was called AUT000201, and the other was DDL920. And both of these drugs activate those parvalbumin interneurons. And guess what? The DDL920 treatment caused complete recovery of motor function in the mice. It was like they had gone through that intensive rehab, but they just got the drug instead.
No kidding. That's incredible.
Yeah, so a drug was able to reproduce the benefits of physical therapy, at least in these mice.
That's huge.
Yeah, it's a big deal, and it has so much potential for future treatments for people.
So like a pill that could help you recover from a stroke, that would be amazing.
Yeah, but of course we have to remember that this is all based on studies in mice.
Right.
So what were some of the limitations of the study?
Well, the first thing is that they mainly used young male mice with a specific kind of movement problem. So we need to do more research to see if it works the same way in older mice, in female mice, and in different types of strokes, because strokes can be very different.
Yeah, so more research is always needed.
Always.
What else?
Well, the researchers used a specific dose and timing for the drugs they tested. So we need to do more research to figure out what the best dose and timing would be for people.
Okay, that makes sense.
And we also need to figure out exactly how these drugs work. Like, what's the mechanism?
Right, what are they doing at the molecular level?
Exactly. And one last thing is that this study focused on mice who had pretty obvious movement problems after their stroke. So we need to see if these findings apply to people who have less severe strokes too.
So the researchers were obviously excited about their findings. What did they say about what this could mean for the future of stroke treatment?
Yeah, they were really optimistic. They think this research could lead to way more effective treatments for stroke, which would be amazing.
That's great.
And they were really excited about the possibility of these drugs being able to help people who can't do regular rehab.
Yeah, because not everyone can participate in those really intensive programs.
Exactly. And they also think this research could lead to new ways to improve recovery by focusing on that neuronal synchronization, getting the brain back in sync.
Yeah, like enhancing the brain's natural ability to heal itself. So we covered a lot of ground in this deep dive. What's one last thought you'd like to leave our listeners with?
I think the biggest thing to think about is how this research could change the way we think about recovering from brain injuries, like stroke. Imagine if a pill could actually help people recover, or even partially replace physical therapy. That would be a game changer.
It would be incredible. And if you're interested in learning more about all this, we encourage you to check out the scientific paper that we talked about, all the other really interesting research happening in neuroscience.
It's a fascinating field.
It really is. Understanding how the brain works is the key to developing new treatments for so many conditions. Thanks for joining us today.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.